The Mind Gala is contributing to the celebration by bringing you an exciting talk by Dr. James Clement about his research on autism spectrum disorder and a possible therapeutic. So, to answer this, let me have the honor of inviting the ingenious Dr. James Clement, our speaker for today's session. His talk will focus on small molecules that restore GABAergic function as potential therapeutics to treat autism spectrum disorder. Dr. Clement works as an associate professor at the JNCASR in Bangalore, India. His lab focuses on intellectual disability and autism spectrum disorder. Please join me in welcoming Dr. Clement with a virtual round of applause. Welcome, Dr. Clement. The floor is now yours.
Thank you very much, Angelica, for the kind introduction. And I'd like to thank Poonam and the other members for the invitation. It's a pleasure to be here virtually to discuss about autism and also highlight some of the recent works that we have done in terms of therapeutics. So, just 1 second. Right. So the therapeutic angle work is done by mainly my PhD student, former PhD student, Vijaya Verma. So I'll be talking about her work when it comes to therapeutics. So my lab is mainly interested in understanding how the genetics and the environment interacts during early stages of development, like newborn babies and also in childhood, to start many neurological disorders such as autism spectrum disorder and intellectual disability. To study autism spectrum disorders or intellectual disability, it is important to understand how these neurons and the neuronal cells function or are during developmental stages that might contribute to the various behavior deficits that we observe in autism spectrum disorders and intellectual disability. It may seem very simple to all of us, but then if you think about it, we were able to feel the hunger at the right time for breakfast, lunch, or dinner, or even snacks, and we were able to sleep at the right time, pretty much plus or minus 30 minutes or so every day, and get up around the same time. And we were able to figure out how to go from one place to another place, even within the same campus, or from the campus to the railway station. And we have a better capability to read, understand, and also imagine. So these things are done by approximately 86 billion neurons and 85 billion non-neuronal cells. It is understood that each neuron makes approximately 10,000 connections with the other neurons that might lead to around 100 trillion connections in the whole brain. So that might imply that the brain is probably the fastest computer in the whole world. So whatever we do in our daily activities from the time we wake up till the time we sleep, and also during sleep, is much easier, and we don't even pay attention to most of the things. For example, when you get up and comb the hair, we don't pay much attention. But if you look at the brain of a newborn baby, they might even have the enough number of neurons that we have, but most of these connections are not even there. The connections are not there, and most of the brain regions are not that active. That's the reason the babies most of the time they sleep, and when they wake up only when they are hungry. But by the time we reach 3 years of age, there's a huge surge in the number of connections. And most of these connections are not stable. They are very flexible. They can form and disappear and again form based on how active we are and how repeatedly we do those things. And when we reach 15 years of age, there's a huge reduction in the number of connections, but still the connections are much more than what we see at birth. So these connections, some of these connections are lost compared to childhood time, and some of these connections become much stronger. The reason they become much stronger is we do the things repeatedly. When we were kids, our parents have told us to do study repeatedly, do things repeatedly, so that we will remember for the exam and other things. And that's true, because unless we do things repeatedly, the connections does not become stronger and they become weak. And like within a few days, we don't remember what we learned. And by the time we reach 18 years, 25 years of age, the ones that are repeatedly activated are maintained, and the ones that are not repeatedly activated or weakly activated, those connections are removed. And this is an example from a postmortem brain. You can see that at birth, there are less number of connections. And in 6 years old child, there's a huge increase in the number of connections. And by the time they reach 14 years of age, the connections are there, but very less compared to a child, but much more than at birth. So this early period of life has a significant effect on how we shape our habits, behavior, our intellectual capabilities, cognitive capabilities, thinking capabilities. All those things are shaped at the very early stages of development. So it is very important that we go out and play as a kid rather than just looking at our laptop and phones. This is true for even um, teenagers, people who are in the mid 20s, early 20s, any age you take, it's very important to do things outside our routine so that our brain, our connections are much more active. But then if you look at the way the baby becomes an adolescent, it takes almost 18 to 20 years. That means the whole brain does not develop at one point, it develops at the different time points. For example, if you look at the senses, that is vision, hearing, and touch, that's the most important thing for the baby. The baby has to see, hear, and feel the touch. Otherwise, it's difficult to sense what is happening outside. Then comes the other parts. So when the baby is born, the number of connections are very less. But then when the brain regions for vision, hearing, and touch are ready, there's a huge increase in the number of connections. So they become very sensitive to even a mildest sound or lightest touch, and even for very faint light sensation, light, they become very hypersensitive. And because the brain is enabling them to adapt very fast, that will allow them to survive when they reach 3 years or 4 years of age. But then after some time, you can notice that the ability to form any connections reduces drastically for vision, hearing, and touch. It comes almost to zero. By the time the baby reaches 1 year or so, the brain region for language, understanding social relationship, talking, everything peaks. But then if you notice here, it does not close completely. I'll explain in a minute why it is. And by the time they reach 3 years or 4 years of age, the brain is ready to understand mathematics, inclusive nature, thinking, reading. That's the reason why we go to school when we are 3 years or 4 years of age. At the time the brain is actually ready for us to learn the higher cognitive things. And by the time we reach teenage, um, our brain is ready to understand what is critical thinking, reflective thinking, consider response. For example, when the parents ask, okay, why did you come late? You think about which question, which answer will put you in less trouble. So those kind of responses, those kind of thinking starts um, when we are teenage. And by the time we reach 20 years or so, end of adolescent, that ability drops. It doesn't completely lost, but the ability is reduced compared to a child. And that's why we get voter's ID and driver's license when we are 18 years of age, because we are able to understand what is right and what is wrong, what to do and what not to do. That's why we are given a voter's license and voter ID card and the driver's license when we are 18 years of age. The reason the language and higher cognition does not completely lost when we attain certain age is because as an adult also you have to live in different cities, so you have to learn a new language, you have to adapt to new cities, new culture, new food, and you have to learn to survive. You will make mistake, we have to adapt to that. So that opportunity is not completely lost, we still adapt to the situation. But the ability to do it and how fast and how quickly we do it is very less compared to a child. And these properties of formation, elimination, and maintenance of those connections are done by hundreds of proteins which are present in the presynaptic region as well as in the postsynaptic region. And if there is a mutation in one of these genes, encoding in one gene encoding any of these proteins can lead to various neurological disorders. The ones are pink are already implicated in autism spectrum disorder. So the gene that my lab is interested in studying is the SYNGAP gene. I will explain to you later what is SYNGAP. And so as I mentioned that a mutation in any one of these genes, including this protein, will lead to abnormal synaptic function, and that can lead to various brain-related disorders, which are highlighted in many different movies. I'm sure many of you might have seen some of these movies which are on autism, not only on autism, but also on memory loss, and also in personal disorders, etc. There are many such movies like that, which you should probably watch to understand what happened to these patients and how do they behave. And it was very well, autism was very well depicted in a movie called My Name is Khan but in his personal life, he doesn't have autism. That's a disclaimer. But if you haven't seen My Name is Khan, I definitely recommend you to watch this movie. It's very well made to make the society understand how people with autism suffer and what do they go through in their daily life. So what happens in the brain of these regions? So according to WHO, they define autism as the lack of ability to learn and adapt to new skills. For example, if you're able to hold a paper for the first time, you don't forget it. You were able to do it continuously. If you learn how to cycle, you were able to continue to do that. But people with autism have those defects. They can't continuously do that. They don't have the ability to learn and apply the new skills, and that usually begins before adulthood with a lasting effect on development, and it is continued during the adult stages. As I mentioned earlier, there is this trajectory followed for a normal brain or for a healthy brain development. But in mutations, what might happen is it can lead to hyperconnections or hyperconnections. So some people might think that, okay, hyperconnections are good, more connections are good. No, the brain needs the optimal number of connections. Anything more or less will lead to abnormal information processing. For example, you may not be able to understand what is 1 plus 1. It will take you months to understand, or you may not even learn. If you look at some of the autistic patients, you will understand that the difficulty in learning the simplest tasks in their everyday activities. And the major challenging question that is facing all the neuroscientists, especially the people who are working on autism, is to enable those children to at least process the information 80 to 90% of a healthy brain, not necessarily 100%, 80 to 90%, so that they can lead an independent life without relying on someone else. And that's one of the questions we are trying to address. And patients with autism spectrum disorder have have reported many behavior issues. One of the major ones is intellectual disability, severe behavior problems, like they may make tantrums, they'll be uh, having some anger issues, and they will be more inclined to spend more time with the objects rather than with the same age group of people. And autism spectrum disorder is very predominant. They have high sensitivity to pain. For example, if you touch a dosa tawa, you will immediately sense it, but they may not sense it for a few seconds to sometimes, or close to a minute. They have eating problems because muscles may not develop properly. They might have gut issues. They have a lot of sleeping problems because of epilepsy and other related issues. Like as I mentioned in my previous slide, the major issue is the current drugs are aimed at treating the comorbidities, the associated symptoms, but not aimed at the main issues, which is the intellectual disability and autism spectrum disorder, because once the connections are formed after that particular critical period, where I showed you the peak, it's difficult to change those connections. It's completely hardwired. It's not that easy. Like, so think about it, how easy it is for you to change one simple habit at this age. It's very, very, very difficult. Not impossible, but extremely difficult to change one particular habit of ours. So because the connections have to be changed, and it's not that easy without having any secondary effects. So most of the drugs are targeting at the minor issues rather than the major issues. So to understand and find a better therapeutics, we use a mouse model called SYNGAP mouse model. And SYNGAP is downstream of NMDA receptor, which is one of the major glutamatergic receptors involved in memory formation. And the yellow one is the AMPA receptors. Again, it is necessary for long-term memory formation. And NMDA has a magnesium block, and magnesium block will prevent the glutamate from allowing the calcium to enter when the glutamate binds to the NMDA receptors. So magnesium has to be removed so that glutamate can bind and perform its function. So in order to remove the magnesium block, the membrane potential of the postsynaptic neuron has to be depolarized, and that is done by the AMPA receptor. So when there is an activity, presynaptic neuron releases glutamate. Glutamate that binds the AMPA allows magnesium to be removed, and that opens the channel, allows calcium to enter. When calcium enters the postsynaptic neuron, it phosphorylates kinase 2 and phosphorylates SYNGAP1. If you notice here, under normal condition, when there is no activity, SYNGAP's function is to suppress RAS activity. Thereby, it suppresses the AMPA receptors from going inside. When there is activity, more RAS activity occurs, and that allows more AMPA receptors to be inserted in the postsynaptic neuron. However, if in case of a mutation, the RAS level inhibition on the RAS is reduced, and that allows more RAS to be synthesized. And without any activity, more AMPA receptors are inserted, and that prevents the neuron from functioning properly because it has to happen when there is activity, but it behaves as if there is an activity, but in fact there is no activity is sensed, and when the neuron is exposed to actual activity, it cannot perform its duty because it's already occupied by the AMPA receptors, and it cannot synthesize new proteins. And synaptic mutation has been has been identified in many regions in the world. So far, around 800 children are affected. It's a little bit old, and most of these mutations are de novo mutations that leads to mutation in the synaptic protein. Children with these mutations have moderate to severe intellectual disability and language impairment, epilepsy, anxiety, and some of them have ataxia. Only heterozygous mutation has been reported in patients. The reason is not from my lab, but earlier studies have shown that the homozygous mice does not survive for more than 2 to 7 days of birth. So it's possible that children with homozygous synaptic mutation may die within a few months, and nobody will ever know why they died, and whether it's because of synaptic mutation or some other mutation. And these are some of the mutations that has been identified so far, and you can see that they are spread all over the SYNGAP gene. And it's so the severity depends on where the mutation is. If it is close to RAS-CAP binding domain, the severity is much higher. If it is in the other domains, then it is much less severe. So the severity changes based on where the mutation is. So how can we study these things in the lab? You can take the human brain, or we can take some animal that has a longer life. We need some animal which has a shorter life, and it can be modified to mimic the human disease or disorder. So we use a mouse model, which is created by Professor Richard Huygenius in America, and it has a heterozygous mutation where a couple of exons are replaced by a neomycin acid. And how do we validate whether this mouse model is a good mouse model, whether it's not only a mouse model, whether it's a Drosophila or zebrafish or C. elegans? How do we know this is a good model to study? So there are three validities to be followed in order to understand whether a particular model can be utilized as a good model to study a disease or a disorder. First one is the face validity, that should resemble the phenotype or the symptoms of the human in the human, and it should recapitulate those phenotypes. And the second one is the concept validity, that should match with the biochemical or the molecular pathways that is reported in the human patients. If these two are fulfilled, that's a really good model to just to understand the disease or the disorder. The last one is the predictive validity. If the model is a good model, then we can test the drugs before it goes for clinical trials, whether this drug can actually restore not only the behavior but also the signaling mechanisms. In some cases, we can restore the signaling mechanisms but not the behavior. When you talk about autism, people are not really interested in what happens to the molecular mechanism. They want to know what happens to the behavior or the phenotype. So it's important to understand that when we study autism, the drug or the compound that we evaluate must rescue the behavior issues and not just the molecular or the biochemical angles. So the question that we asked is whether this synaptic heterozygous mutation affects the normal synaptic transmission. That means they don't have any proper activity. How do they behave? How do the pre-synapse and the post-synapse behave? To understand the question, we use electrophysiology, which is a fundamental to understand the function and dysfunction of neurons and their systems. That will also explain how the communication happens between synapses in terms of disease or under pharmacological conditions. And this is our electrophysiology week. And this is a mouse brain. You can appreciate how small it is, but it pretty much mimics what humans go through. They may not be able to speak like us, but they do pretty much everything that we do. And this is a neuron in the cortex, and these are the glass electrodes that we use to patch neuron. And to ask a question on basis and transmission and how the synaptic mutation might affect the basis and transmission, we usually take the region called the hippocampus because it has nice boundaries, and because hippocampus is necessary for memory formation, and also it has this inbuilt GPS for navigation. It has multiple functions. You can read about it a lot on the any Google websites. And what they have done is in this particular region is called dendrite gyrus. So hippocampus has three regions: dendrite gyrus, CA3, and the CA1. We focus mainly on the dendrite gyrus because that's the main entry point of information into the hippocampus. The message comes from the prefrontal part in the infrared cortex, and we have activated those axons and recorded the postsynaptic responses, and the responses typically looks like this. We have two negative going currents. The smaller one is the fiber body. It is the summated action population action potentials, and you see a huge negative response. So that is your excitatory postsynaptic neuron. So this is like your mouth, and this is more like your ears. And for a given action potential, we should see a corresponding increase or a corresponding response from the postsynapse. It can, if it is increases or decreases, that means the basis and the transmission is affected because of the drug or because of the mutation. So when we look at it from different age groups, and I forgot to mention earlier that the developmental time point of hippocampus is 3 weeks. I was telling you about 18 years, etc. But the equivalent of that is 3 weeks in the hippocampus. So we took different age groups, 1 week, 2 week, and 3 weeks old, and also in adults. And you can see here, so what we have plotted here is the action potential on the x-axis, which is the fiber volley, versus the postsynaptic response, which is the field APHD on the y-axis. And you can see the blue ones are the wild type, and the red ones are the synaptic mutant mice. And you can see here that in 2 weeks old, there's a huge increase in the responses, which is comparable with the 3 weeks old and the adult stages. The reason why it is happening in 2 weeks is I haven't shown the data, but we know that from the mRNA expression, synaptic mRNA expression reaches the peak when in 2 week old hippocampal brain. But then after that, it kind of declines and stabilizes. So that tells us that the major impact of synaptic protein is in 2 weeks to 3 weeks of age, and that is reflected here. And because the synaptic protein is not functioning properly, it is disrupting how the connections are formed during developmental time point. And as I said earlier, there is that increase is matching with the adult group. So what it tells us is the brain is maturing. Instead of 18 years in humans, it's happening in 10 years. So everything is advanced, and that's going to be detrimental for the brain development. And when we look at the structure, for example, the spines, I will tell you what is spines in the next slide. That is also maturing in P14 to 16-day-old hippocampal region. And as I mentioned earlier, SYNGAP suppresses the AMPA receptor insertion, and if SYNGAP is not there, it should allow more AMPA to be inserted, and that also we have um, identified and reported, and that is also happening at 2 to 3 weeks of age. And so these experiments tells us the brain is maturing much earlier than when it should happen. Instead of 3 weeks, it's happening in 2 weeks. If you think about humans, instead of 18 years, it might be happening when they're 10 years, and that's going to disrupt everything for the rest of the life because the brain requires optimal connections at the right time. It should not happen before or it should happen later. If it is 6 months, it should happen in that 6 months. It should not happen before that or after that. As I mentioned, if the electrophysiological properties are changing, the structure might also change. The way we store our memories are in the small structures called spines, and this is a dendrite. Each of these spine they look like these thorns in the rose plant. I hope everybody has seen a rose plant, and the thorns, they appear like that, and they come in different shapes. And they come in different shapes. And here you can see these spines are properly arranged, and they have different shapes in a healthy adult. But these three are from patients with autism spectrum disorder, and you can see how the structures are completely disrupted and abnormal. So if you have such structures, we can't have this kind of proper communication between pre-synapse and the post-synapse, and that will affect how the information is processed in the brain. It's impossible to learn new things and remember new things. And because of the mutation, if you look at the neuron, and again, this is in the visual cortex, and the developmental time point is 60 days and not 3 weeks. And this is wild type, this is mutant, this is wild type, this is SYNGAP heterozygous mutant mice. And if you look at this SYNGAP mutation in 21 day and compare it with the 68-day-old, you hardly see any difference. They're pretty much similar. So that again confirms the way it branches, the volume it occupies in the brain, also happening much earlier. Again, that's going to disturb how the connections are removed and maintained during developmental time point, and that process of elimination and formation of new connections is extremely important for a child. And if that process is disrupted, it's going to affect the capability to learn any new things and how to behave in a party, how to behave in a classroom. Everything will be completely disrupted. Again, this recapitulate the spine structures that I shown earlier, and also matches with the physiological recordings I showed you earlier, that everything is shifted by a week or so in the mutation as a result of the mutation compared to the wild type. So if these kind of mutation time point being shifted, it might affect how the excitation and inhibition. So excitation helps you to tell what is your name, and inhibition controls the activity of a neuron. It will not be activated unless there is a requirement, like somebody asking what is your name, how to do those things. For example, think about epilepsy. People cannot control it. So that means there's an imbalance between excitation and inhibition. If we are seeing such kind of abnormal activity, abnormal change in the presynaptic to postsynaptic function, and also the volume it occupies, change in the spine structures and the density, it might lead to abnormal neuronal activity. So I'm going to show you a short video, and this is again hippocampus. It was captured live, and we're going to stimulate one specific neuron using ammonite glutamate. And when the neuron is activated, generally the activity is restricted to that particular region. Well, and it doesn't spread properly to the neighboring region. That means excitation inhibition is balanced. Whereas in the mutant, you can see what happens is that activity spreads to the neighboring region, similar to at a level similar to the local area or the region like dendrite gyrus. So it doesn't restrict itself to dendrite gyrus, but it goes all the way to the other region. So that will disrupt how the information is being processed in the brain. So what we concluded is early maturation of synapses causes excitation inhibition imbalance. So the next question we asked is whether this imbalance can cause epileptic seizures. So what we have done is, I mentioned earlier that these neuronal activity are hypersensitive to sudden to light or sound. So if this excitation inhibition balance is not maintained, that might lead to behavior deficits and also epileptic seizures. So this is a wild type mice. This is a mutant mice, and it has a white paint on the tail, just in case if you lose track of it. And we have given a 120 decibel sound. It's like somebody dropping a 10-liter cooker right next to you. But then we recover very fast. We startle, but we recover. We don't have epileptic seizures. But people with um, abnormal connections, they can lead to epileptic seizures. For example, when we have migraine, we are sensitive to even the smartphone light or even sound. That is because of increased sensitivity to external stimuli. So you can see here when the alarm goes on, you can see that mouse is running around properly. It couldn't control, and wildlife gets scared. But then you can see what happens to the heterozygous mutant mice, that the epilepsy becomes so bad, it dies. I have actually fast forwarded for the sake of time. So it kind of runs around, and it couldn't control what is happening, and the wild type mice is pretty much terrified of what is happening to the other mice, and then the synaptic mice dies, and we have lost 50% of the mice from epileptic seizures. If seizure is happening and excitation imbalance is affected, it might lead to various cognitive, emotional, and social behavior deficits. So how do we study those things? So here, to study sociability, we use three chambers. Like humans, mice also tend to spend more time with the other mice and not with objects, and they're able to distinguish between old mice and new mice. For example, if this is an old mice and you put a new mice, they will spend more time with a new mice. For example, when a new person joins in the lab, you're curious to know who are they, where are they from, what do they like, you try to spend more time with them to understand who they are. And the mice also does that. And mice with synaptic mutation or any other uh, mutation implicated in autism, they do have such deficits. They will spend more time with the object or alone. They never spend more time with the other mice, and also they do have a tendency to be less anxious and hyperactive. So we evaluate that in the open field. When you see um, rats running around outside the house, they don't tend to come to where the light is. They tend to run close to the wall where there is a protection. So these lab mice also they do that, and you can see that they spend more time on the walls. They spend less time in the center. They do go to the center, but less time there because they know that if they go to the center, they're exposed and they can be caught by the predators. Whereas in mouse with mutation implicated in autism, they don't have anxiety, so they spend more time in the center compared to the healthy mice, and they also cover more distance compared to the healthy mice. And how do we study memory? So this is a well-known paradigm called Maury's water mice, and you can see a platform at the bottom of the screen, and that is not completely visible. That's just below the surface of the water. So the mouse runs around and finds the platform. The first time they will take some longer time, maybe a minute or so, but then the second time, third time onwards, they take a few seconds to reach there. And due to the lack of time, I'm just fast forwarding it. You can see even after 16-20 seconds, it hasn't found the platform. Eventually, it finds it, but then this is what the um, autistic mice does. They can't remember where the platform is. They don't recollect where the platform is. And when we did those experiments, not only from my lab, my postdoc lab, and also from other labs, they have shown that these heterozygous mutant mice prefer to be socially isolated. They are less anxious and they are susceptible to epileptic seizures. They have increased locomotor activity and they do have long-term and working memory deficits. Going back to the initial question, challenging question, can we restore the um, memory deficits by targeting GABA? The reason I'm talking about GABA is there are many studies have shown that GABA is a better therapeutic target than any other neurons or neuronal cells in the brain because it has less restriction on how the neurons are, are functioning. So using another mouse model of autism, which is the Fragile X model, it's a well-known gene which is implicated in autism. So what this lab has done is they've taken a region called somatosensory cortex. That's a region that corresponds to the whiskers in the mouse, and their developmental time point is 7 to 9 days. And when they have given a bumetalide, which blocks the chloride cotransporters, which regulates whether GABA is inhibitory or excitatory, and I'm not going to talk about more on that. So when they targeted the GABAergic function in the wild type, it does not have any effect. And whereas you can see that in the mutant mice, the membrane potential is much depolarized compared to the wild type. But when they gave the drug during the critical period, it is restored. That is easy. That's why the clinical trials fail because by the time we take a child to the doctor, most of the brain regions are developed. So what is the point of finding a drug that works only during the critical period? We need to find a drug that works when when the brain is almost developed or just developed. Right. So when they tried that, which is after the critical period in green color, you can see that it is not restoring like it restored when they gave it to the early stages of development. So again, this is one of the reasons why most of the drugs fail because they don't test when the brain is actually developed. So what we have done is when we were doing some of these experiments, a paper came out showing that a biocompound was able to restore this GABAergic activity and bring back the um, membrane potential back to the normal level when administered in a cell line of Rett syndrome. And we had this compound called 6-bio, which Dr. Ravi and I have published for in Parkinson's also. And we know it crosses the blood-brain barrier. So Ravi and I discussed that why don't we test it in the autism mice? We have this, we do see these deficits in SYNGAP mutation. Why don't we check it out? And Dr. Sridhar is our organic chemist who was kind enough to synthesize tons of these 6-bio. And the fantastic student I had, Vijaya, she was working very hard and we test injected in 3 different age groups. The first age group is 10 to 16. That's a critical time point. That's why everyone does it. And if it works, it's fantastic, but it's not going to be that useful for the patients. So we use 3 different age groups. The one is during critical period. And the next question is, do we have to administer from the critical period till the time the person is alive? And the most challenging question is, if we inject after the brain is developed, imagine the hippocampus is 21 days, so most of the experiments were targeted at the hippocampus. And if we administer after the brain is developed or that particular region is developed, can it still restore the function at least partially? Partially is good enough. The patient doesn't have to be dependent on someone else for everyday activities. So I'm not going to show the show the group 1 and group 2 because that's not really exciting because tons of labs have done it. So I'm going to show only the results from group 3, where we have administered the compound from 30 days to 80 days, sorry, not 60, 80 days. And that is pretty much equivalent to like giving a drug when the kid is 8 years old. And we have evaluated through these 3 behavior experiments and also looked at the LTP. Just to recapitulate, we have done the open field to understand the hyperactivity or the locomotor activity of the mice and also the anxiety level. And we looked at the memory test. We done the memory test by subjecting them to the novel objects. They're trained on two similar objects, and 24 hours later, new object is introduced, and the mouse will spend more time with the new object. And we have done the social novelty test, mouse between object, old mouse versus the new mouse. And when we have done the open field test, like others are published, in synaptic mutant mice, without the drug, they tend to be more active. They tend to cover more distance. And when we gave the drug, we can see that it is pretty much um, the same as a vehicle. So motor locomotor activity was not restored in when we administered. It kind of partially restored, but not completely restored in the mutant mice when we administered after the brain was that region considered to be developed, but it did restore when we gave it to the critical period of development. And when we check the anxiety level, the synaptic heterozygous mice had was less anxious. It was entering to the center of the open field too many times. But when we gave the drug, it partially restored it when we gave the compound after the brain region was supposed to be developed. So that was really encouraging. And when we checked for social interaction, so this is the empty versus the stranger, and you can see that they're not able to distinguish it. But when we gave the compound, the mind was able to distinguish between the old object and object versus the mice, and it was able to spend more time with the particular mouse. And when we checked for the older mouse versus the new mouse, again that was disrupted in the synaptic mice, but that was restored when we gave the compound. So remember, these are all done when the brain region was considered to be developed. So again, this was really encouraging results. And the most important question was, can we restore the memory? Here we look for novel object recognition. So here anything close to 25 or 30 and below is considered to be no memory. Here you can see that they're not able to remember compared to the wild type, and the memory was restored in the drug administered synaptic heterozygous mutant mice. That means they're able to remember what they were able to remember to distinguish between the old object and the new object, and they tend to have epileptic seizures, like I showed you in the video. Patients do have different levels of epilepsy. Some of them do have absence seizures, and that prevents them from sleeping properly. And when we gave the drug, you can see that the threshold for epileptic seizures was restored to the healthy mice. So this, what it tells you here is that the threshold is much lower to induce epilepsy. That means even a mild, like for migraine, when we have migraine, we are hypersensitive to light or sound. That means our threshold to that particular sensory system is low. So that's what it means here, that the ability to induce epilepsy has a much lower threshold compared to the wild type level, and that threshold was restored to the wild type level in the synaptic mutation. So in summary of the behavior results, we have the 3 age groups, and you can see that motor function was corrected during critical period, but not in the adult stages. That means we had to treat the motor function. We have to start a little bit earlier. Anxiety was corrected. Partial, partial correction is also good because that will help them to somewhat lead a normal life. And normal object recognition was corrected even during the post-critical period of development. So this is the very important age group. These many studies have shown with multiple compounds. When we test them here, they all fail. This is the most important group. And with social interaction and social preference, it was corrected. So social preference was not corrected maybe because the connections are not even there for certain um, processing of information. And epilepsy was corrected at this particular age group. We can't do much earlier because it's difficult to assess, and also we won't get the ethical approval. And in terms of physiology, it is again the synaptic plasticity was restored when we gave it from the critical period and also when we gave it after the critical period of development. So in summary, since it's a general talk, brain is a complex organ and it needs an appropriate or optimal development for everyday activities like combing the hair, brushing our teeth, having a shower, and etc. And intellectual disability and autism spectrum disorder are due to mutations in genes encoding proteins that are important for neuronal function as well as for non-neuronal function. And in synaptic heterozygous mutation is one of the causes of intellectual disability and autism spectrum disorder, and that roughly affects 1% of the population. And still, not many studies have evaluated for mutations in SYNGAP in patients. So we don't know really how many patients do have SYNGAP mutations. And SYNGAP mutation leads to early maturation of connections, and that causes a delay in development in children. So we call it as milestone in children. For example, if they have to walk when they're start crawling when they're 8 months of age, that is a milestone. So those milestones are disrupted because of the abnormal neural connections as a result of the heterozygous mutations, allowing the brain to mature much earlier. It has to happen at the right time. If it doesn't happen at the right time, we don't get the right proteins for the right function, and everything is messed up. And above all, how do we maintain these things even as an adult and also when we're above 50 years of age? So one of the major issues is lack of nutritious diet. People are not eating healthy food, like they spend more on Coke and other things, and also eating burgers and not so healthy food. And the other important thing is the exercise. We have to do physical exercise at least 3 times a week, and people don't do those things. So that's going to disrupt how these connections are maintained, they are formed. And it's important to have a good sleep. If you don't have a good sleep, by spending more time on phones after 11 p.m. or midnight and staying awake, that sleep is going to disrupt how these connections are formed and maintained. And whatever you learned the previous day, you may not be able to remember it for the next day and also in the coming weeks. If you want to have a good memory and want to be more productive, you need to have a good sleep. And I just finished with a couple of quotes. A typical neuron makes about 10,000 connections. That means there are as many connections in a single cubic meter of a brain tissue as there are stars in the Milky Way. And Francis Crick said that there is no study that is more vital to the man or a woman than the study of his or her own brain. The reason is who we are and what we think about entirely depends on how our brain functions. If that is disrupted, who we are and how we function in a given day is completely affected, and that leads to lots of problems. None of these will be possible without the wonderful group that I have in my lab. These are all the people who have been there before, and also the current group, and my wonderful collaborators and the funding agencies from DSD, DBT, and also mainly from the institute. Thank you very much for your attention. I'll be happy to answer the questions.
Thank you so much for the very insightful talk, Dr. Clement. We now open up the floor to questions. So please, you all can type your questions or just turn on your mics.
Hi, I have a question.
Yeah, sure.
So I think a couple of days ago or a week ago, there was recent findings on microplastics being detected in the blood. I was wondering, what do you think the effects would be if it passes through the blood-brain barrier, and would it have an effect on autism specifically?
Yeah, that's a good question. Yes, it will definitely impact how the brain functions. It's going to affect how the synaptic plasticity occurs in the brain. And if you think about even us, we will have a drastic effect on our capabilities to remember things, and it might lead to loss of memory much faster than we anticipate. But in terms of autism, if the pregnant lady is taking lots of microplastics, unknowingly from food and water, that might enter the baby, and that's definitely going to cause some mutations. And if it is something to do with the protein that regulates synaptic function, that can lead to autism. So the answer is yes, it can have a huge impact.
Okay, thank you.
Anyone else?
Hello, sir.
Yeah.
Yeah.
Hello.
Hi.
Sir, it's a wonderful talk. This is Dr. Kumaresan. I have a doubt,
Hi.
like I just want to clarify. Uh,
Sure.
uh, so I have done PhD on autism in environmental model. And before that, I worked with autistic kids almost a year. Like, I wonder, like, the autistic children, do you think they have really the memory problem, or we say it's like sameness because they don't want to change from the routine? Because
Right.
I handle some few kids, like the learning is like once, yeah, they are slow learner.
Yeah.
But once they learn, they are very good in memory, actually.
Yes. Yes. In some
So
angles, yes. Yes.
yeah, so I just wanted to just clarify with your
Yeah.
these
So
things
what
because
happens is
you
there
are
are two
saying,
factors here.
yeah,
So there are two factors. One of them is the memory formation capability that is definitely slow, depending on the
okay,
mutation. But in severe mutation, sometimes what you have seen doesn't happen to many patients, right? It's in severe
okay,
cases. So what you're saying is maybe like more like a mild case of autism spectrum disorder that we have also seen where they can remember. So what is happening is because of the connections are restricted and they're not that flexible when they're supposed to be flexible,
yeah,
and that prevents them from learning anything. If you want to eat with your hands, you have to learn. If you want to walk, you have to learn. So everything depends on how
yeah,
we learn and remember and form the synaptic connections. For walking, it has to form between the motor cortex and the other regions, right? So those connections can be disrupted when they're supposed to be functioning properly, right? So that's one thing. And then to recollect memories, for example, what is your name, where are you, how to behave in a certain situation, those are recollection of memories. So that also can also be disrupted because of abnormal excitation and inhibition. Okay,
okay,
so these are the two different factors that might combine together and prevent them to act or function appropriately, and they might take some time depending on how severe the mutation is.
okay. Sir, how much the this kind of mutation seen in human subjects are like?
Yeah, so it comes to like 3% close to all over the world. If you talk
Okay,
about I'm not talking only about SYNGAP mutation. I would say
okay,
it's between 3 to 4%.
okay, okay, sir. Okay. And I had one
Right?
more
Even in
doubt,
India,
like,
we have lots of people with autism spectrum disorder. Like sometimes I go to NIMHANS and I talk
okay,
to clinicians. They give me the numbers. It's enormous compared to the population.
oh, okay, okay, sir. And you got some seizure activity in your that mutant mice, right, sir?
True, true.
So, like, is all the mutant shows that seizure activity, or it's like some percentage?
So we have only one mutant of mouse model, and all of the
Okay.
mice do show that. But from the patient studies, from whatever I have interacted and read,
Hmm.
majority of them do have epileptic seizures.
Okay,
Not
but
necessarily all of
literature
them, but
says like
90. Mhm.
30% of the autistic children do have the seizure, not all like.
Well, so like I said, that can vary depending on which mutation we're talking about. There are 1,200 genes that are implicated in autism,
Okay.
right? So it depends
Yeah,
on
yeah,
which gene we are
definitely.
talking about and where is the
Oh,
mutation.
okay.
And in SYNGAP, pretty much 80% and above do have epileptic seizures.
Okay.
Even the EEG is not proper.
Okay. Yeah, I have
They may not,
because I
for example, they might not exhibit epileptic seizures, but when you do the EEG recording, which keeps them awake, right, because of the brain waves are not proper, and when we do the EEG, you can see the abnormal activities that matches with the epileptic seizures that we see in mice.
okay. Okay, thank
Yeah.
you.
Good evening, sir. I have
Yeah,
one
good
question.
evening.
Good evening. Sir, I just wanted to ask, is there any stress difference with respect to autistic people and normal people?
So you're asking, if I understand correctly, you're asking whether the stress level is different between patients with
Yes,
autism
yes.
and normal people. Yes, they do tend to have, because they tend to have more, exhibit more stress compared to normal people, because they don't know how to adapt and accept. They
Okay.
prefer, for example, like earlier person was asking the question, they have the sameness. They want to follow the same thing. They don't want to deviate from that. Whereas we are trained to adapt to a situation. So when they deviate, that stresses them like anything. Even you move one object from the usual place to a different place, they get stressed like anything. Right. So the stress level is completely at a lower threshold compared to the healthy adult or healthy child.
Okay, because I have seen some people, so their reflex action is are so fast than the normal people.
Hmm. So you're saying that the reflex is much faster to the normal people.
Kind of.
Is
So
it okay? So I haven't seen that much. Even mice also we haven't seen. Like I interact with some of the kids and talk clinicians also. We haven't seen. Could be some exceptions.
okay, sir.
Right.
No
So
problem. Thank you.
Anyone else?
Um, Actually, if there are no more questions from audience, I would like to ask one. First of all, thanks for the wonderful talk.
thank you.
So you showed one graph with critical periods of development in
Hmm.
brain, right? So
Yes.
in that one, I was wondering that if due to some reasons that critical window is missed, say due to a mutation or a developmental lack of nutrition or something, is that like permanently lost chance, or is there some way that window that can be restored, so there is some hope for autistic children to
So
revive?
it's not completely lost. It's just that the ability becomes much slower once they lost that critical period. The
Mm-hmm.
challenge is to reopen the critical period. So that's where it becomes a problem, because when you want to reopen the critical period, you don't want to disturb lots of connections or even the synaptic function. We want to have less disruption to whatever they have learned so that
Mm-hmm.
they can learn new things. So that's where the drugs fail most of the time, right? So the ability is like complete, not completely 100% lost, but they have only 10% chance, 10 to 15% chance
I see.
after they cross the period.
So how difficult slash easy it is to reopen this critical window period? Is that dependent on us one or two genes, or is that a lot of things working together?
So for example, if you take one specific receptor, mGluas, they might function differently in the post-synapse and also pre-synapse, right? And they may function differently in striatum, differently in motor cortex. So the function slightly changes depending on which region we're talking about. And and the thing is, if you have a drug that's targeting a specific type of receptor, then it might disrupt how that receptor functions in a different region. So it becomes a huge challenge. So that's the reason people think GABA might be a much better bet, because GABAergic function is pretty much uniform throughout the brain. And for example, NMDA also does that. But then how do we change the um, we, if you target NMDA, we may not be able to open up the synaptic function properly, right? So it's not that easy. It's going to be extremely difficult, because once you have to remove the unwanted spines, in the sense like the ones which are less active, and enable new spines to be formed based on the external stimuli. So that's going to be very challenging. People have tried gene therapy. Most of them have actually failed for example, in mice data, and drugs also most of them have failed because they're not able to remove the less active ones and enable the new formations. And people have targeted mTOR, that also not that successful. So it's a very complicated process, and it's extremely challenging. We don't want the neurons to die also. We want to preserve those neurons, but just enable the connections, makes it feasible for the connections to be removed and formed, form new connections. So it's a very complicated procedure.
Yeah, I see. Thanks.
Okay, so with no more questions coming in, we call it a day. We now invite Dr. Poonam Thakur, the co-founder of the Mind Gala initiative, to address the gathering and deliver the vote of thanks.
Yeah, so sorry for my scratchy voice. I'm a bit under the weather, but I would really like to thank Dr. James Shilaya for showing up with us tonight and talking about autism. And since we are celebrating Autism Awareness Month this time, we thought this would be a very timely theme for our session. And I would also
you.
really like to thank all the audience members who have stayed us in later part of their evening and remained here and asked very insightful questions. And in the next month, we would have a lot of more events coming up for you. And not just a webinar, stay tuned. We have more surprises coming for you next month. Please follow us at our Twitter handle, Mind Gala, and keep updated with our more offerings that we'll be announcing very, very soon to all of you. So thank you once again, all of you, for joining us today. Thank you and see you next month.
Thank you so very much, Poonam. Thank you. Bye.
Thanks
Thank
so
you.
much, Poonam. So we thank you all for being an amazing audience. The Mind Gala would be back with a series of exciting initiatives around the year. So make sure to tune in to our social media handles to know more. Until then, stay safe and protected. Thank you once again. You all can now leave the meeting. Thank you so much, Dr. Clement.
Thank you so much.
Bye,
Bye.
sir.
Bye.